The article I chose is The Unusual Nose and Brain of the Star-Nosed Mole by Catania and Cass. When looking at a photo of a star-nosed mole, it is easy to assume that the nose is for smelling, but in actuality the nose is a touch organ. This organ consists of 22 fleshy and mobile appendages that fan out around the nostrils and allow the star-nosed mole to explore their environment. What is of interest to the researchers is how information from this unique nose is processed within the brain. The researchers used several different methods to test this hypothesis: physiological recordings to map the cerebral cortex, scanning microscopy to look at the skin surface of the nose, and recording neurons in the cortex to understand how the star-nosed mole responds to stimulation. The researchers began by examining the sensory organs on the nose and by observing how the mole uses its nose to explore the environment. Results from the physiological recordings raised some interesting questions. Within the mole's cortex, the researchers found that the star-nosed mole has a relatively large somatosensory area. This is the area specialized for processing the sense of touch. This large somatosensory area has a morphologically distinct pattern of subdivisions. Each one is related to one of the appendages of the nose. These findings can offer insight into how the brain areas subdivide during development and reveals new information concerning the relationship between the peripheral sense organs and the cortical areas that receive input from these sense organs. The researchers go on to explain foraging behavior in the star-nosed mole. When a star-nosed mole is hungry, it uses its nose to search for food. The mole moves its nose in constant motion, touching different places as it moves forward searching for food. This movement is extremely fast. The mole lifts its nose and touches different areas 10 or more times a second. It also seems that different appendages are used differently when food is encountered. We'll explore this more later. Here is a video clip taken from the BBC's life documentary series. As you can see, the star-nosed mole is exploring his or her environment using the appendages on the nose. I think it is important to be able to see this in motion, so that's why I added this clip. As you can see in the image, there's 22 separate and distinct appendages. The appendages consist of 11 pairs that are symmetric across the midline. In the photo, the appendages have been labeled 1 through 11. Appendages 1 through 9 are the longest appendages and account for most of the surface area of the nose. Initial contact with prey occurs with one of these appendages. After contact, the nose immediately shifts so that the 11th appendage can make repeated contact with the food before it is eaten. Once prey has been located, the 11th appendage is referred for detailed tactile exploration. This entire process can take place in less than half a second for small prey, which we will see in the following video. To fully understand how the appendages work, it is best to see it in action. As you can see in this video, the star-nosed mole is using different appendages to find and eat its prey. As previously mentioned, another method the researchers used was scanning electron microscopy. They used this method to study the specialized skin on the surface of the nose. In low-power scanning images, as seen in photo A and B, it appears that the appendages are covered with a honeycomb pattern of small epidural papillae. Each of these papillae is an Eimer's organ. An Eimer's organ is a touch organ that contains a complex of several types of sensory receptors. The star-nosed mole has approximately 25,000 Eimer's organs over its 22 appendages. An important note of interest to the researchers is that the Eimer's organs are not distributed continuously across the entire skin surface. At the base of each appendage, there is a small strip of skin that does not have any Eimer's organs. This strip separates the receptor array for the appendage from the receptor array on neighboring appendages. Another area of interest to the researchers was how this nose evolved, particularly how the nose of the star-nosed mole is far more delicate than most other mammalian skin surfaces. It seems that the star-nosed mole evolved this nose by exploiting a previously unused food niche. When the Eimer's organs of the star-nosed mole were compared to relatives that live in drier soil, it turned out that the size and form of the Eimer's organs correlates to the particular habitat of the species. The star-nosed mole lives in wetlands and digs in mud. Moles in drier soil have larger Eimer's organs, while the star-nosed mole has a smaller, more delicate Eimer's organs with a thin epidermis. Here's where things get pretty confusing. When Catania and Cass studied the cortex of the star-nosed mole, they found the nasal anatomy is an extremely well-developed tactile sensory system that the behavior of the star-nosed mole revolves around. The researchers recorded neurons in the cortex, which revealed that a large portion of the cortex contains neurons that respond to stimulation of the nose. After recordings were made, they examined the somatosensory area of the cortex by sectioning the tissue parallel to the cortical surface and staining for the enzyme cytochrome oxidase, which revealed the relative metabolic activity of different brain areas. What was interesting from this research on the cortex is that the star-nosed mole is unique in that all three representations of the nose are visible in histochemical stains as a series of cortical markers that reflect the peripheral sensory sheets on the appendages. The star-nosed mole is the only species with such patent markers in 3 different cortical areas. These areas form a cortical processing network. The neurons that represent each appendage in each somatosensory area are directly interconnected with one another. The image above is showing the peripheral nerve branches from the 11 nasal appendages compared with the cortex. The nerve branches are the darkly stained clusters in the center of the tissue. Image B is an example of the somatosensory cortex stained with cytochrome oxidase. The areas with different intensities of cytochrome oxidase staining have different cortical connection patterns with other neurons, so they may have different functions in the processing network. This is one of the figures I found particularly fascinating. This is an example of cortical magnification, which is of interest to the researchers. The illustration at the top shows the correct anatomical proportions, while the lower illustration is showing the anatomy relative to each other in the somatosensory cortex. There are many more nerve fibers in the nose than any other body part. As you can see, the forelimb also has a large representation in the cortex. The researchers think that this suggests that the forelimb is an important sensory surface, may be used for interpreting soil composition and density, and maybe even detecting ground vibrations produced by approaching predators. What's interesting to note is that the 11th appendage is much larger than originally projected from the number of fibers innervating the 11th appendage. After further investigation, the researchers concluded that the area of the cortex representing presenting each appendage is not proportional to its corresponding innervation density, but rather it is related to its preferential use in feeding behavior. The appendages most heavily used during feeding, 1, 9, 10, and 11, have the most cortex per nerve fiber. In conclusion, Catania and Cass found the star-nosed mole has a specialized nose and brain which provides researchers with a unique view into the organization of the somatosensory system in the mammals. Much to the surprise to the researchers in the study, the representation of the nose in the cortex is a lot larger than expected, and this ended up being due to the behavioral significance of the appendage rather than the number of Eimer's organs or the number of nerve fibers. These findings raise many questions. How do preferentially magnified cortical areas develop? Are they the result of greater relative activity during early development or throughout the lifetime of the mole, or are they genetically determined? Further research on the star-nosed mole could provide insight into questions concerning brain organization.